thanks a lot. So I'm John Koestel. I'm working at Agroscope in Switzerland, close to Zurich. And basically, we have the X-ray images that Statis wants to have, but we don't have the nice soil hydraulic properties going with it, and we also don't have the opportunity to model it. So what I will show you now is simply results from experiments that aim to elucidate how soil macrostructure imaged with X-ray CT evolves with time and what drivers in it are. And I want to highlight collaborators who did most of the work. That was Yuting Fu, who is doing a postdoc with us, and Sharon Chebet, who is doing her PhD. And then Maria Vorkauf and Florian Walder, who also are senior researchers here. So basically, to move forward. Okay, the motivation already Nick and Statis have given, so I won't spend much time on that, only to say that it is more or less, there is some ideas about factors influencing soil structure evolution, but what factors are more or less important is not so clear. And it's also not so clear what the frequency and amplitude of the changes in soil structure are, because these are rarely measured. And so the aim of these experiments that I want to present here is to simply to investigate the soil structure evolution and to relate it to possible drivers. And the main tool with this is something that we call soil structure incubation. So first of all, a short primer in X-ray imaging. Probably most of you already know this. With X-ray imaging, you basically can map the 3D distribution of densities, and it's encoded traditionally like this, that you have bright colors, that is high density. So in this horizontal cross-section here that you see to the left, these are kind of gravel and sandstones. And black, that is the lightest density, that would be air-filled pores. Then you have intermediate values. These dark gray, this corresponds to particulate organic matter, and also water would show up like this if you basically would see that in the image. So these images always were taken at matrix potentials where all visible pores would be air-filled. So you hopefully don't see that. Okay, and the light gray, that corresponds to the soil matrix. This is basically regions where you cannot resolve either particulate organic matter or minerals or air-filled or water-filled pores, but you have a mix of all of them, and that's why they show up in this kind of rather light grayish value, grayscale value. Okay, and to make clear that actually these are 3D images, so we have this information in 3 dimensions. So the left, you see this grayscale image of a block cut out of an X-ray image, and to the right, you see the air-filled pores that were extracted from it. So, and just because we are talking to a group that is kind of working on pedotransfer functions, and the most classical pedotransfer functions found soil hydraulic properties. Here kind of a relationship between KS, which is kind of notoriously difficult to predict from classical soil properties, but it gets better if you have the soil structure. So if you have undisturbed soil, what seems to work quite well, not perfectly, but better than everything that I know, is that the saturated hydraulic conductivity is proportional to the square of the critical pore diameter. And the squared critical pore, the critical pore diameter would be shown in red here, so that would correspond to the largest sphere you could fit through the pore system from top to bottom. So there's one work that we did together with Attila Nemes when I was still working in Uppsala in Nick's group. that is shown here. And there was a similar work done in the group in Halle at the U of Z with Steffen Schlüter that also found that basically that for undisturbed soil, the critical pore diameter is best correlated with Ks. But if you have an aggregate bed or seed bed, then it's rather the macroporosity that is best correlated with Ks, because then you don't have kind of this one passage that kind of dominates the flow through the whole sample. But you have several flow paths which are equally important. Okay, and finally, to kind of now get you set up completely. So what is meant with soil structure incubation is meant the following approach. Here you see 2D vertical cross sections. So first you sample soil, then you sieve the soil, then you repack it. Here on the left, you see how it looks if you repack it very loosely, just by kind of adding layer by aggregates and shaking a bit. And then you install this sample in the field and simply wait. And after a time, you excavate it and do another X-ray image. And then you can see basically on the right, you see the same soil sample and an X-ray image and a vertical cross-section through it 3 months later after it was installed as this seedbed or this seedbed-like aggregate assembly. And then you can, of course, repeat until you run out of funding or whatever other boundary you hit, and kind of have then a time series of soil structure evolution in the one and the same individual soil sample. Okay, so the first experiment to show that was a soil structure incubation experiment with size exclusion. And the aim was to see what role a macro and mesofauna and roots would have in shaping soil structure. Okay, and basically what we did, we sampled soils from, well, actually it was Chebet who did that. She was sampling soil from grassland, sieved it to 4 millimeters, repacked it loosely, and she had 24 replicates for each treatment. And she had 3 treatments, one without any additional mesh to this kind of, it's cutlery holders for dishwashers, which have very nice plastic that is very durable and have openings which are more or less 12 millimeters across. Yeah, I should also say these samples, these are maybe 12 centimeters in height and approximately 10 centimeters in diameters. And these are the same kind of samples which I will show you now in the rest of the talk. Okay, so we had one treatment without any mesh. Then we had a treatment where we had a 1 millimeter diameter mesh-sized mesh, nylon mesh, and one with a 40 micron. And the idea was that basically in the coarse mesh treatment, we would exclude all the macrofauna, while in the no mesh, we would have access to invertebrate macrofauna at least, so not to mice. And so on, but at least to earthworms. And in the fine mesh treatment, we would exclude all meso and macrofauna and only have basically microbes, fungi, maybe some nematodes, and no roots as well. And basically, this was then installed in the field. The meshes were tried to make tight so that nothing could kind of, everything had to go through the mesh, and we also aimed at kind of establishing hydraulic contact with the surrounding soil. And after 14 months, basically from December to the next year in February, it looked like this. Like this. So you have now 2D cross sections on the top with the no mesh, where you can see, okay, you see an earthworm. I hope you can see my mouse. Here to the upper right, and in this first replicate, you see an earthworm up, and you see other biopores which are characterized by roundish cylindrical shapes. And you see also that the original assembly of aggregates in the two treatments, or especially in the treatments with no mesh applied, has fused considerably, whereas kind of if you have the fine mesh, actually the original structure is still like it was in the beginning. It is fused a bit more, but apart from this settling, the structures did not seem to have changed awfully much. And in the coarse mesh treatment, you have some intermediate case. So you can see the same thing as well in the 3D rendering, where now you see the pore space here, and the pores are color-coded by diameter. So the brighter the colors, the larger the diameter. And you see, of course, if you don't apply any mesh, then you have the largest biopore diameters. And if you apply the fine mesh with a spacing of 40 micrometers, then you don't have any imaged biopores. And you basically have an interaggregate pore space. Okay, so I thought I'd show you also the results for the critical pore diameter. Obviously, for the fine mesh, you still have this kind of aggregate packing, so you cannot apply the critical pore diameter as a measure for the hydraulic conductivity or the saturated hydraulic conductivity. But you see that the more you allow access of roots and fauna, the larger the variability gets, or variance gets. Not so much maybe the average value, but the variability. Okay, so the summary of this first part would be that, okay, if you have abiotic and microbial factors combined, so this was not enough to alter the soil macrostructure in 14 months. So it is often, you often read that kind of there are by capillary forces, the aggregates would fuse and the soil matrix was fused together. And that may be the case, but it was apparently not happening very quickly. And if soil biology, larger soil biology, macrofauna and mesofauna has access, this process gets much, becomes much quicker. And basically, interestingly, the soil pore structures in the coarse mesh, when only macrofauna were kind of restricted, were then similar to the ones where there was no restriction. But they were completely different to the ones with the fine mesh, where there still was the original structure. And basically, one takeaway, if you angle it to the KS, was that if you have excess of larger fauna and roots, then you have a kind of a larger variance in the critical pore diameter and supposedly also in KS. So the next experiment was actually already carried out in Sweden. Just in the last years I was working there, and then in the first years I had already been working at Agroscope. So it is a soil structure incubation experiments in the garden soil, where you, maybe some of you know, there's a paper published on the same garden soil structure incubation in 2019, I think. And this was basically a follow-up that I did start without funding to learn kind of how this kind of would work. And I was mainly interested in if to see how large the fluctuations were in these macropore structures, if there were seasonal dynamics, how long the macropores, the individual macropores that I would see in the individual columns were existing, and if roots, if they decay, would leave root channels. So basically what I did, so I had 9 columns. Again, it was this cutlery holder with the same dimensions as before. Initially, I tried to seed these plots with plants with different root characteristics quite drastically, so even carrots or Jerusalem artichoke. And then actually I had started the experiment in summer 2018, and there was the soil columns were repacked with loose aggregates after hand plowing and seed preparation. And then every half a year a scan was taken, actually even during Corona, but not the year after, because then Nick will probably remember there was a long time where the scanner was not working in Uppsala. And so basically the last 4 scans I did when I was visiting Uppsala, yeah, in the go. Okay, so the first result, so how did the macroporosity and bioporosity change? And how did the particulate organic matter, which is largely composed of the roots, change with time? And you see here kind of the 9 different columns, and you see, well, yeah, there's quite some difference and there's quite some fluctuation. And to come to the questions, the hypothesis that we had when starting this experiment is kind of we were first thinking that the root network, the soil pore network would evolve to some kind of equilibrium, but find out no, that's not the case. Actually, it evolves with the plants that are growing there and the root biomass that they have in the soil. So you could see here kind of the variations, and actually it's not so much the average value that changes, but again, the variance in here, for example, the particulate organic matter that is imaged. You can see it increases statically with time, and with it also the porosity is increasing. And you can also see that actually there is some kind of a seasonal trend. For example, from autumn, where you have kind of a more variability in the particulate organic matter content, to spring, there is a decay probably of, yeah, not probably, there is a decay of the particulate organic matter. So it is eaten up over the winter year. And then it's created anew during the next growing season. Or, yeah, that's how it is. And well, and if you go there, yeah, if you look at the POM fraction, you see the POM fraction really varies a lot depending on the individual soil column. So this column number 4, this is the one where the Jerusalem artichoke was growing, and there you had a lot of kind of below ground immature biomass kind of created, which was then eaten up subsequently. And there were other columns, for example, this column number 1, where not so much was growing. And there you had much, you had less kind of particulate organic matter, but you also had less macropores than. You see kind of these seasonal changes also in the porosity. And you also see that sometimes these changes with season in the POM or porosity, they are not really consistent. So here you see a steady increase in POM, and here you have also in the last years in the porosity, you see, okay, you don't have this pattern of having basically a decrease in image porosity over the growing seasons, probably because the roots are growing, taking up space. You don't have that always, right? In the last year, you don't have it. And we're not sure why yet. Okay, next hypothesis. So what is the lifetime of individual large biopores? So here you can see in yellow, the biopores that persisted over the winter from 2019 to 2020. These are 2 example columns. But you see then over the next growing season, almost all of them were kind of reworked and kind of were not existing anymore. There's a bit more persisting in the column to the right, but kind of the largest part is simply kind of restructured. So biopores really, at least if there are kind of most of them that we observed have a very short lifetime of maybe 6 months at most, probably on average rather less. Unless in this column where we had an ant nest that remained active, and there we had the most stable pore, macropore structure over all time. So the lifetime of biopores also vary with origin. And the kind of the hypothesis number 3, that roots would leave biopores after decay, equivalent to the diameter, that was found to mostly not be true, right? There's kind of one old root here that is just caught in the process of decaying, and then, yeah, okay, there seems to be biopore left behind when it decayed, but other roots, and that was in the majority of cases we observed this kind of, they decayed and were eaten up, and you have a lot of kind of earthworm spill or other spill around it. And here on the bottom also you see, I mean, the root that was formerly there just leaves no pore at all. Maybe here on the bottom you see a bit pore that is left, but it's much larger than the original root has been. So that that tells us that, okay, if you have large roots, probably I mean, we cannot prove that, but it makes a lot of sense to me. They are very attractive. They provide a lot of food, and they attract mesofauna that feeds on them, and they reshape the pore structure around them. And finally, what about the critical pore diameter? That's shown here in gray, and you see, yeah, it varies kind of wildly. And we didn't focus on the critical pore diameter in this manuscript that is about to be submitted. So I don't know what the trend is, but you can see, okay, so in the same soil sample, if you incubated, the soil saturated soil hydrologic conductivity should change really drastically. If you accept that the critical pore diameter or the square of the critical pore diameter is a good measure to predict it, or good indicator to predict it. Okay. So the image pore networks evolved with plants. As a summary here, then seasonal fluctuations were discernible, but depended on the plants growing on the plot. The average lifetimes of large biopores was shorter than a year, except in the presence of ants. And the decaying roots may have left root channels after decay, but their lifetime then of these root channels was very, very short, clearly shorter than 6 months. And well, and the critical pore diameter and the saturated hydraulic conductivity probably varied widely in the individual columns. And then I hope I have time for the last experiments. This is kind of now funded soil structure incubation time series experiment in the Soil Structure Observatory, which is a long-term field experiment here in the Reckenholz that was initiated by Thomas Keller and was original soil compaction experiment. And we didn't look at the compacted treatment, but we looked at the chemically established bare fallow, where basically the soil structure was not changed. Basically plants were killed with glyphosate and so on to keep the soil bare, and compared it to the pore structure evolution in a permanent grassland. Again, soil was sampled, was sieved to 4 millimeter in diameter, repacked loosely. So we had per treatment, so there are 3 blocks in these experiments. In each block, we had 4 replicates. That means per treatment, we had, or per block, we had 4 pseudo-replicates, and basically 12 kind of pseudo-replicates per treatment. And these soil columns were excavated every 3 months for 2 years, from November 2023 to November 2025. And they were always X-rayed and then reinstalled into the soil again. And this is how basically the pore system looked in the bare fallow. You see in the beginning, it's again some interaggregate pore space, but already after 3 months, you have a lot of kind of biopores established. And actually, we are not sure what is making these biopores. So a lot of parallel tubes often seen that go very straight down in the soil and then stop abruptly. So we know that on this bare fallow, there were a lot of ground nesting bees. So it could be that this is the kind of organism that makes this burrows, but yeah, we're not yet sure, and we're trying to find out. If you compare it to a column that is from the grassland, you see, okay, you also have a lot of biopores, but they look completely different, right? They're more irregular, they have larger diameter, and then on the bottom you see these large caverns showing up, and again, these are ants that have been moving in here. Okay, so we can, with the X-ray images, we can distinguish between bioporosity and non-bioporosity, whereas I have to say as a caveat, this distinction is not very exact because it depends on the algorithm that is used to calculate it, but it's approximately kind of a measure of how many biopores we have in the imaged pore space. And you can see that often in the bare fallow, the fraction of the biopores in the whole imaged soil is even larger than in the permanent grassland. Yeah, which was quite surprising to us. So mostly it was surprising that there is so much biological activity. And then if you look at the average values of thickness, that's basically the pore diameters that have been imaged, you see that yeah, it's a bit bigger, larger in the grassland, but it's not significant. But it becomes significant later in the observation period, and probably that's our guess. It's because of the ant burrows that establish in the grassland, but not in the bare fallow. In terms of macroporosity, you see a clear difference between grassland soil and bare fallow. And yeah, and you also see kind of what is very striking is again that the variability in macroporosity in between the columns is much larger in the presence of more biological activity, so in the grassland, than in the bare fallow, where you have relatively little variance in the macroporosity. And well, then if you focus or look on the lower right, you see a connectivity measure. That's a measure of how well the image pores are connected to each other. And also here you see much larger connectivities in the permanent grassland. And again, much larger variances than in the bare fallow. Only in the critical pore diameter, which is then serves again as an indicator for saturated heterodic conductivity, you see no difference between the treatments. You also see large variability, but this is this time in both treatments relatively large. And to give a preliminary summary, so macroporosity and connectivity of the image pore networks much larger in the grassland, but there's still surprisingly large biological activity in the bare fallow, and even the fraction of biopores is as large or even larger than in the grassland. However, if you look at the type of biopores, you see that the biopores in the bare fallows look different than in the grassland, indicating that the the architects of these biopores were also different. And of course, you cannot have any biopores created by roots because they are killed off supposedly quick enough to create root channels. Yeah, again, you see that there's much smaller variability in the bear follow in the measures that are shown you, and most measures that we measured, but the critical pore diameter, and which was not affected by the land use. Yeah, and then I thought maybe, yeah, do we see something like an equilibrium if you look here at the macroporosity that has established then maybe after the first year? So this is rather speculation, but well, we are hoping we can continue with this experiment for a while, and then we will know more about that. So to the overall conclusions, and now the last slide. So from these experiments, kind of one take-home message is that the macropore network dynamics and also the macropores found in the soil depend strongly on the local plants and the macrofaunal activity. And you must not assume that you would reach a structural equilibrium in the soil unless you also have the same plants grown there and the same macrofauna active there. So if this changes and evolves, kind of in the extreme case to forest, you evolve also with your soil structure, which is what I dare to claim, but it looks like this. Yeah. So there is seasonal dynamics in the macropore network, but it seems to be complex, also depending on the plants, local plants and macrofauna. And yeah, right. Yeah, you have more biological activity, you have more spatial and temporal variance in the macropore network. But, sorry to jump over these 2 points here, if you especially average the critical pore diameter here, it doesn't look so bad, huh? It seems it stays relatively constant, huh? So maybe Statis is not so wrong with saying that kind of, yeah, do we really need this kind of change? Or well, sure, it's only the critical pore diameter and the saturated hydraulic conductivity, but it also goes in this direction. So maybe there is a lot of variance and temporal change in the soil properties, but maybe a lot of it is also canceled out by spatial variation. Okay, and then the other two things can be taken away here is kind of that large macropores appear to be short-lived in these experiments that we looked at, and large root channels or root channels of large roots appear to be extremely short-lived. So with that, thank you, and I hope I stayed more or less in time.